Hello everyone, we're so glad you've joined us for this live webinar, Uncertainty, Choice, and Dopamine. I am Soon Pham of LabRoots and I'll be moderating today's session. Today's educational web seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. Let's get started. You can pose questions to the speaker during the presentation at any time while they're fresh in your mind. To do so, simply type them in the Q&A box, which will open when you click the green Q&A button at the lower left of the presentation window. Questions will be answered after the presentation. To enlarge the slide window, click on the screen icon in the lower right-hand corner of the slide window. If you experience any technical problems seeing or hearing this presentation, click on the support button found at the top right of the presentation window, or report your problem by clicking on the green Q&A button lower left. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located at the bottom left-hand corner of your web page and follow the process of obtaining your credits. I would like now to present today's speaker, Dr. Stan Floresco. Dr. Stan Floresco is a professor of psychology and member of the Brain Research Center at the University of British Columbia and a fellow of the American College of Neuropsychopharmacology. With over 90 peer-reviewed publications, Dr. Floresco specializes in research on behavioral and neurophysiological approaches to study neurocircuits within the dopamine system that facilitate higher-order cognitive functions such as cognitive flexibility and cost-benefit decision-making. He's also interested in how dysfunction in these circuits may relate to psychiatric disease. In 2010, Dr. Floresco was awarded the American Psychological Association's Early Career Award. I will now turn it over to Dr. Floresco for his presentation.
Well, thank you to LabRoots for inviting me to give this talk, and for all of you joining us online, thank you. And for those of you who are watching this on demand later, glad you could make it. So today I'd like to talk to you about some work my colleagues and I have been focusing on over the last 10 years or so now, looking at different neural circuits within the dopamine system and how they regulate certain aspects of reward seeking within the context of risk-reward decision-making. Now, life routinely presents us with situations where we have to choose between different options that may yield us different types of reward. These rewards can differ in terms of their subjective value, choosing between two things we like. We just happen to like one more than the other. Godiva's chocolate versus Hershey's, craft beer versus Budweiser, what have you. Or they can differ in terms of their objective value, basically more versus less reward. And all things being equal, we tend to like larger versus smaller rewards. But the decisions that we'll wrestle with, that give us pause, are when these larger or better rewards are associated with some sort of cost that can diminish the subjective value of objectively larger rewards. And these costs can take a variety of forms. Sometimes we may have to work a little harder or wait a little longer to obtain the things that we prefer. Or, as will be the focus of my talk, sometimes these larger or better rewards might be associated with some sort of uncertainty or risk, and we might not always get rewarded for choosing a certain course of action. Now, these types of decisions are particularly tricky because here you have to do the long-term cost-benefit math and try to ascertain, well, maybe I won't always get rewarded if I choose a certain course of action, but will it be more beneficial for me in the long term if I play a little more risky or a little more conservative? Now, studies on the neural basis of risk-reward decision-making really find their roots in the mid-90s with the pioneering work of Antonio Damasio and Antoine Bachar, who devised a novel task we now refer to as the Iowa gambling task that was designed to tap into real-world decision-making in terms of uncertainty, rewards, costs, and punishments. Now, since those original studies, with the advent of new tasks, use of functional imaging, and with looking at different patient populations, we now know that different aspects of risk-reward decision-making are mediated by distributed neural circuits in the brain that link different regions of the frontal lobes, the amygdala, which resides in the temporal lobes, and the ventral portion of the striatum, also known as the nucleus accumbens. And one thing that all these regions have in common, in addition to their own interconnectivity, is that they're all also connected with the mesocortical limbic dopamine system, which suggests that dopamine may also play a role in these functions. So we have known for nearly half a century that dopamine plays some role in reward, although admittedly, those of us who study this molecule can't always agree exactly what its function is. With respect to dopamine neurons, one thing we know about these cells is that their electrical activity is particularly sensitive to situations involving the presence or absence of expected or unexpected rewards. So if one records the activity of dopamine neurons in an awake behaving animal, and they present them with either rewards or cues associated with those rewards, one sees a characteristic pattern of activity. So if we take an animal and present it with an unexpected reward, what that does is that causes these dopamine neurons to fire a brief phasic burst in activity, to increase their activity. Whereas if we now start giving the animal some information about those rewards, present a certain cue that reliably predicts that the reward is coming, eventually these dopamine neurons actually start to fire in response to that cue associated with the reward, but not necessarily the reward itself. On the other hand, let's say we present the animal with a cue associated with that reward, but then when the animal is expecting the reward, we don't actually deliver it. Well, in that case, we actually see a suppression in dopamine neuron activity, what we refer to sometimes as a phasic dip. And so these increases and decreases in dopamine neuron activity associated with expected or unexpected rewards or non-rewarded events are thought to encode what are referred to as reward prediction errors, which may help an animal organize its behavior and change its actions when it's not getting what it expects to do, or promote continued action towards getting these rewards. Now, it seems that dopamine neurons are also particularly sensitive to situations involving reward uncertainty, because we can change the scenario that I just described to you here. Now we can present the animal with a variety of different cues. Some cues are never associated with reward. There's a 0% chance of getting a reward when that cue is presented, while another cue might always be associated with reward, 100% chance. And then there may be some cues which only deliver rewards a certain portion of the time, 25, 50, 75% of the time. And these are seminal data from Wolfram Schultz's group. And what they found in this study is that the activity of dopamine neurons in response to these different cues was maximal in response to the cues that presented the maximal amount of reward uncertainty. So that dopamine neuron activity was much greater when the animal was presented with a cue that suggested it might get a reward 50% of the time, when you compare that to cues that either always predicted reward or that never predicted reward. So this suggests that the entire dopamine system is particularly ticklish when the organism is not quite sure whether or not they're going to get a reward. Now, studies in humans have been monumental in helping us identify some of the different systems that may be involved in using this information to guide decision-making. But they don't really allow us the experimental control we need in order to understand the mechanisms underlying the processes that help guide our behavior when we have to choose between different rewards of different probabilities. So we really need to turn to the animal model to help understand how these different systems may interact and what their specific roles may be in guiding risk-reward decision-making. And so work by our group have been using an assay that is referred to as a probabilistic discounting task, where we get animals to choose between different types of rewards, varying in terms of their magnitude and their relative certainty. So I'm going to spend a bit of time discussing this assay because most of the data I'll be showing you today was using this basic type of task. So this task is conducted in an operant chamber, and the way this game works is on any given trial, we present the animal with two options, two levers. If the animal chooses what we call the small certain lever, it always gets this smaller one pellet reward. So this is the safe Canadian government savings bond option. You're not always going to get that much of a return, but you're always guaranteed some return on your investment. Now, on the other hand, on this trial, the animal might choose what we call the large risky lever. Here, the animal may or may not get the larger 4 pellet reward. And the probability of the animal getting this reward changes in a systematic manner over blocks of trials within a daily session. And so the way we typically run this game is at the start of the session, the odds of getting the reward are very high, either 100 or 50%. And then as the session progresses, the odds get leaner and leaner. to 50, 25, and then 12.5% chance of the animal getting that large reward. And changes in these reward probabilities are signaled by blocks of forced choice trials before every block of free choice trials. So first, in every block, we force the animal to choose either one of the levers. And then this way it can ascertain what the relative probability of them obtaining that reward is. Then we give them a period of free choice trials where both levers are presented. And now they have to choose, based on the information we gave them, which lever they prefer, which one they think is the better option for them. And if you take a look at this graph here, after about 20-something days of training, animals become relatively rational decision makers on this task, choosing that risky option more when reward probabilities are high, and then choosing it less when the reward probabilities are lower. So in all of the studies I'll be talking about today, animals were well trained on this game. So by the time we did our neural manipulations, they were experienced players. And within any group of animals, we have some animals that tend to be more risky and some animals that tend to play more conservative. And so we would use a within subjects design to see how, when we alter different nodes of dopamine circuitry, how does that alter the way they play the game on that particular day. And so in our initial studies, looking at how dopamine might be involved, we simply did systemic treatments with different drugs that interfere with dopamine transmission. One of the first things we found is that if we increase dopamine release by giving a drug like amphetamine, we get this marked increase in risky choice. Animals were much slower to update the changes in their decision bias, and they persisted in playing that risky option, even though the odds were getting poorer and poorer. On the other hand, if we reduced dopamine transmission by giving a drug that blocks dopamine receptors, such as fluphenfixol, we see the opposite change in behavior. Now animals actually played much more conservative. They were risk averse. They didn't like choosing that large risky option and would rather choose that smaller but safer reward, suggesting that dopamine tone in the brain helps bias choice towards these larger uncertain rewards. So these data told us that dopamine, somewhere in the brain, might be helping modify these decision biases when animals choose between risky versus certain options. So over the last few years, we systematically targeted different nodes within the dopamine system to try to understand what their functional contribution to these problems are. And in these experiments, before we started playing with dopamine, we just simply used reversible inactivations of different nuclei, where we would temporarily shut down neural activity in these different regions to kind of ascertain what role do they play in guiding decisions. What do they bring to the table in terms of helping to solve these types of problems? What I'm showing you here is a cartoon showing some of the neural circuits that we've looked at over the last few years and showing their intrinsic connectivity. So you can see one nucleus, the basolateral amygdala, which resides in the temporal lobes and is important for emotional processing. It sends projections to the nucleus accumbens. The basolateral amygdala also shares reciprocal connections with the medial prefrontal cortex in the frontal lobes. And this cortical region also sends projections to the striatum. So I'm going to summarize about 6 years worth of data in the next 90 seconds or so, just to show you what our studies with inactivations have shown, what these different regions contribute. So what we found is that one circuit linking the basolateral amygdala to the nucleus accumbens seems to play a key role in biasing choice towards these larger uncertain rewards. Because if we inactivate either structure bilaterally, or if we disconnect communication between these two structures, we get a marked decrease in risky choice, similar to what we see when we block dopamine receptors. So this region is the one that is promoting choice of these larger uncertain rewards. For those of you who play poker, this is the part of the brain that lights up when you have a pair of kings in your hands and is saying, go all in, chase after that big reward. But these urges to play more risky are tempered by the frontal lobes. And this particular region of the prefrontal cortex, the medial region, analogous to area 32 of the anterior cingulate in humans, seems to play a more cognitive role in helping adjust decision biases and keeping track of actions and outcomes over multiple trials to help ascertain what the relative beneficial action is What are the relative odds between these different options? And one of the reasons I say that is because if we inactivate the medial prefrontal cortex, when the odds are initially good at the start of the session and then get poor as the session progresses, we see an increase in risky choice. Animals keep choosing that risky option, even though it's not as beneficial to do so. However, if we change the game a little bit and have another group of animals where the odds are initially poor at the start of the session, start at 12.5%, and then get progressively better, inactivation of the prefrontal cortex actually causes the opposite pattern of effect. Animals are slower to update their decision biases and overall choose less risky. So what we think the prefrontal cortex does in this situation is it monitors changes in reward contingencies that in turn helps the animal change its behavior, alter its decision biases when things are changing in its environment. So one thing we were really interested in seeing, well, how do some of these circuits talk to each other? Are they working independently of each other, or are they actually cooperating, sending information back and forth to help refine these decision biases? And we were particularly interested in understanding whether the amygdala and the prefrontal cortex interact directly to guide decision-making. Now, when we do a bilateral inactivation, shutting down both regions of a nucleus in one brain, that will tell you that nucleus is involved in a certain behavior, but it won't tell you who it's talking to, what other nuclei might be interacting with. But one way we can delineate the routes of information transfer between brain regions is by using a relatively old-school traditional technique called an asymmetrical or disconnection inactivation procedure. Now, the rationale behind this procedure rests on two assumptions. First, it assumes that for any given behavior, information may be transmitted from one structure, in this example, structure A, to another structure, structure B, on both sides of the brain in parallel. Furthermore, it assumes that a unilateral inactivation of any structure by itself should have no great effect on behavior, because you will still have a contralateral circuit available on the other side of the brain. So what we do in this asymmetrical disconnection procedure is on one side of the brain, we use a unilateral inactivation of the output structure, and then we combine that with a contralateral inactivation of the receiving structure downstream. So what this selectively does is it prevents communication between this A and B circuit on both sides of the brain. So in this example, on the left side of the brain, structure B can't process information from structure A because we have that inactivation upstream. On the right side of the brain, structure A is intact, but now structure B can't process any information being transmitted from structure A because structure B has been inactivated. So if we need selective communication in this circuit, then this type of asymmetrical disconnection should disrupt behavior. But let's say that structure A is not talking to structure B. Let's say it's talking to another structure, say structure C. Well, in this case, we would not expect a big effect on behavior because we would still have a free circuit on one side of the brain. So we use this procedure to ascertain whether the amygdala and the prefrontal cortex form a functional circuit to help guide decision-making. And so as you can see here, the data, when we just infused saline as a control procedure, animals showed rational pattern of decision-making, choosing the larger reward more when the odds were good and choosing it less when the odds were bad. Now, when we conducted an ipsilateral inactivation, so when we disconnected communication between the amygdala and the prefrontal cortex only on one side of the brain, we got no change in behavior. But it's when we disconnected communication on both sides of the brain between these structures where we saw profound change. Now, to be quite honest, we weren't sure what was going to happen here, because as you may recall, when we bilaterally inactivated either structure, either the amygdala or the prefrontal cortex, we actually got opposing effects on behavior. When we inactivated the prefrontal cortex, animals chose more risky and were slower to update their decision biases. Whereas when we inactivated the amygdala, animals played more conservative. So we weren't sure if a disconnection might just cancel both of these effects out, or we might get a different pattern of behavior. Well, it turns out when we selectively disrupt prefrontal amygdala communications, we get a prefrontal-like phenotype. Again, animals were more risky. They were slower to update their behavior. They kept choosing that risky option, even though the odds were getting poorer and poorer over the course of the session, suggesting that these two regions are communicating to help refine decision bias and modify action selection when there are changes in reward contingencies. Now, one thing we do in these types of studies is we do a more fine, detailed analysis of the behavior. Try to understand, well, why exactly are animals playing more or less risky? And so we do a choice-by-choice analysis to see, well, are they playing more risky because maybe they like the rewards better? So we can look at win-stay behavior, looking at the probability of them choosing the risky option after playing risky on the previous trial and winning. And in this case, that process didn't seem to be effective. Instead, the reason the animals seem to be playing more risky is that they were less sensitive to losses. We saw a decrease in what we term their lose-shift behavior. So normally, when an animal plays risky and loses on a trial, doesn't get that reward, we see them shift to the more conservative option on the next trial about 30 to 40% of the time. When we prevented the amygdala and the prefrontal cortex from talking to each other, we saw a marked decrease in this lose-shift behavior, as if the animals either were unaware or didn't care that they were losing more as the session progresses, so they kept playing risky. Now, as I told you, this pathway is reciprocally connected, and even though I don't have time to go into the details of the data, we have done some follow-up experiments where we have identified that this effect, where we see a disruption in choice behavior when we disconnect the prefrontal cortex and the amygdala, is actually mediated through a descending pathway, where the prefrontal cortex seems to exert top-down control over the amygdala. So these signals seem to originate in the frontal lobes, and then they send a mediating influence over choice behavior that is normally driven by amygdala ventral striatal circuits. So when we take these data and kind of integrate it into a kind of model circuit diagram that we are building to help understand how these different regions interact to guide behavior, well, when the animal's choosing between different options, we have one circuit, this subcortical, more visceral circuit, linking the amygdala and the ventral striatum that is biasing choice towards these larger uncertain rewards. As if to say, we want it! Go after that big reward. But these urges are tempered by the frontal lobes, whereas they exert top-down control over the amygdala, and the prefrontal lobes keep track of actions and outcomes over time, ascertain that, hey, this particular course of action is not yielding us as much reward as it used to. Perhaps we should change our strategy. Perhaps we should maybe play a little more conservative. And then it uses its influence to suppress or mediate these urges to chase after those big rewards, driven by these subcortical circuits. And so this is a common theme we see in emotional regulation as well, as we have these subcortical systems that are driving emotional responses, but they are modified and tempered by different regions of the frontal lobe. So now that we had an idea of some of the key hardware that the brain uses in terms of solving these problems, we were interested in seeing some of the software that might be involved in these processes, namely how dopamine may play a role. And in these studies, what we did was simple, straightforward psychopharmacological studies where we would administer small volumes of drugs that interfered with dopamine transmission in our two key terminal regions here, the medial prefrontal cortex and the nucleus accumbens. Now, in one study, we targeted the mesoaccumbens pathway, and the results here were relatively straightforward. One thing I can tell you is that when we targeted different dopamine receptor subtypes, so there are two key receptor subtypes we were interested in, the dopamine D2 receptor and the D1 receptor. Whereas when we blocked D2 receptors in the accumbens, we saw no real change in choice behavior. Now, these doses were behaviorally active. Animals were slower to make choices. They moved less in the chamber, but disrupting D2 receptor transmission didn't seem to affect their general pattern of choice behavior. Instead, the big player in the accumbens seemed to be the D1 receptor, because when we blocked these receptors, we saw a decrease in risky choice. Animals chose that risky option much less. And in this instance, the reason they were playing more conservative, the reason they were more risk-averse, was this manipulation caused an increased sensitivity to losses. So when they played risky and they lost, they were now more likely to switch to the conservative option on the next trial, in opposition to what we saw when we disrupted prefrontal amygdala circuitry. So these data suggest that within the ventral striatum, or the nucleus accumbens, the D1 receptor plays a key role in mitigating sensitivity to losses and promoting choice towards these larger uncertain rewards. Now, in the prefrontal cortex, the story was a little bit more complicated. Here we also targeted these two key dopamine receptor subtypes. And when we blocked dopamine D2 receptors here, we saw an increase in risky choice, very similar to what we see when we inactivate the prefrontal cortex. And so when we took these receptors out of the equation, when dopamine was not allowed to signal through these receptors, animals again persisted in choosing that risky option, even though the odds were changing. So we interpret these data to mean that one function of dopamine D2 receptors in the frontal lobes may facilitate exploration of different options when rewards changes. Because when we inactivate or when we block these receptors, animals are slower to start exploring new options when the reward probabilities decrease over time. Now, what we were particularly taken back was, when we blocked dopamine D1 receptors, we actually saw the opposite change in behavior from when we blocked D2 receptors. Animals, again, were more risk-averse, similar to what we saw when we blocked these receptors in the accumbens. And again, they were more sensitive to losses. If they played risky and they lost after we blocked D1 receptors, they were more likely to switch to that conservative options. So we interpret this to mean that D1 receptors are playing a distinct yet complementary role to what D2 receptors are doing in the prefrontal cortex. And these receptors may facilitate exploitation of currently favorable circumstances. So the idea is, when the animal plays risky and loses, activity at D1 receptor sites may help bridge the gap between rewarded and non-rewarded actions, as if to facilitate the ability of the system to say, hey, you didn't win on this particular trial, but keep at it, because it may be more profitable in the long term. Don't be so disgruntled because you didn't win on this trial. It might be better for you to keep at this risky option. So, as a psychologist, we love dissociations. We love it when two brain systems are showing opposite changes in behavior. But this is a weird one when you think about it, because the brain doesn't release a D1 or D2 receptor agonist. It releases dopamine, which would be expected to stimulate both receptors. So how does the same neurotransmitter system, working in the same brain region, mediate different patterns of behavior, seemingly opposite patterns of behavior, just by working on different receptors? So for that, our eyes were kind of reopened to the idea in some recent studies that came out in 2012 that showed that there actually appeared to be separate populations of projection neurons within the prefrontal cortex that only express D1 or only express D2 receptors. So we have what appears to be separate circuits that are only modulated by one receptor or the other. And so this may be how dopamine may be able to exert differential effects when we take one receptor out of the equation or the other, by working on separate microcircuits. Now, we follow these experiments up with some other studies trying to see whether these different circuits may have different projection targets. And I won't have time to show you the data today, but what it looks to be going on is that we may have one circuit of a number of prefrontal neurons that contain D2 receptors that project to the amygdala, that mediate one pattern of choice, that mediate exploration of new options, that facilitate changes in decision biases. Whereas another circuit, made up of neurons that contain primarily D1 receptors, may be interacting with a separate subcortical nucleus, in this case the ventral striatum, to promote exploitation of currently favorable circumstances. So those of us who study dopamine and are hoping to find a grand unified theory of its function, well, these data suggest that our task is a little bit harder. Because even when we're looking at just one single brain region, it seems that within a particular brain region, dopamine may be doing different things and facilitating different cognitive operations and patterns of behavior by working on distinct subcircuits or ensembles of neurons. And these neurons can differ in terms of the specific dopamine receptors that they express and their particular projection targets. So as we continue to try to understand how dopamine may be involved in cognition and behavior, we're going to have to account for the fact that we have separate groups of cells, even within one particular brain region, that may be promoting quite different patterns of behavior. So these studies showed us that dopamine clearly played an important role in facilitating modifications in decision biases and promoting choice of larger, riskier rewards. We were particularly interested in seeing, well, how do changes in dopamine release relate to changes in the animal's behavior? Do dopamine levels just go up and stay up, or are there dynamic fluctuations in dopamine transmission? Before we get to those studies, I just want to integrate these dopamine data here with this emerging circuit that we're talking about. So we have our prefrontal amygdala striatal circuits, and within this circuit, dopamine seems to be doing different things in different brain regions and acting on different dopamine receptors. D1 receptors in both the prefrontal cortex and the nucleus accumbens seem to reduce the sensitivity to non-rewarded actions and promote and maintain choice biases. So it helps an organism keep that eye on the prize. Maybe you didn't win this time, but keep at it because it might be more profitable in the long term. Whereas D2 receptors in the prefrontal cortex seem to facilitate changes in decision biases and promote exploration of new options. And within the prefrontal cortex, as I said, D1 and D2 receptors may mediate different aspects of reward seeking by acting on distinct ensembles of prefrontal projection neurons that may have distinct subcortical projection targets. So how do dopamine levels change when animals are engaging in these behaviors? Well, to do this, we collaborate with my former PhD supervisor, Tony Phillips, and his long-term research associate, Soyan An, and we used in vivo microdialysis, which permitted us to look at fluctuations in tonic dopamine levels in these different terminal regions while animals were actually performing and playing the game. And in these experiments, animals were again well-trained on the task, and on our test day, we would implant them with microdialysis probes in either the prefrontal cortex or the nucleus accumbens, so we could see how dopamine levels changed over different phases of the task. And in this task, we were able to use a sampling interval of 7 minutes. Why do I bring that up? Well, this relatively short sampling interval would allow us to look at changes in dopamine release during the forced choice portion of each block. So when animals were only presented with one lever, so didn't actually have to make a decision, just had to respond on whatever option we gave them, and then compare that to changes in dopamine release during the free choice portions of the sessions, where they actually had to make a choice. And in these experiments, we had separate groups of animals. Some were trained where the odds initially were good at the start of the session and then got poor, changed from 100% to 12.5%. And other groups of animals where the odds were initially poor at the start of the session and then got better, started at 12.5% and progressively increased. Now, we needed a key control experiment in these tasks because anyone who knows anything about dopamine is just that the mere consumption of food is enough to turn this system on. So if we see changes in dopamine release when rats are engaged in decision making, we needed to ascertain, well, how much of this signal is driven by them actually having to make decisions and evaluations versus the mere consumption of food. So in these experiments, we included a key yoked food control experiment. Animals in these groups, they didn't have to press any levers. They didn't have to make any decisions. What happened with them is on any given trial, every 40 seconds, the house lights came on, and they would receive either 4 pellets, 1 pellet, or 0 pellets. And the scheduling and amount of rewards they received during these sessions were matched identically to animals that had actually performed the task. So here we could control for the amount and the scheduling of food that they received. So in these groups, animals were just basically receiving food for free. Didn't have to work for their dinner. They would just get this food, but they got it on the exact same schedule as an animal that actually had to work for its supper and had to make decisions in terms of how having to maximize its rewards. So here I'll start with what we saw in the prefrontal cortex. And one thing I can tell you is that when we looked at animals where the odds went from good to bad and compare that when the odds went from bad to good, we saw mirror image profiles of each other. And we were able to combine the data, and when we plot these data in terms of changes in prefrontal dopamine release as a function of reward probability, you can see in these green circles here that dopamine levels were higher when the probability of obtaining that larger reward was higher, and then they were lower when the odds of getting these 4 pellets were lower. So one of the first things we noticed about this change in the dopamine signal is that it didn't really map on very well to changes in the animal's behavior. What these white circles are showing you here is how often animals actually chose the large risky option over the course of the session. And as you can see, the change in choice behavior didn't really mirror the change in the dopamine signal. Instead, what it looked like was the change in prefrontal dopamine levels and the signals from the dopamine system that the frontal lobes were receiving mapped on very well to changes in how much food, how much reward the animal was receiving over time. What these triangles are showing you is the relative amount of reward pellets animals received over the session. And as you can see here, the slope of the change in the amount of food they received, looking in that curve, was practically identical to changes in the amount of dopamine that was being released over these comparable sessions. And what's more, when we looked at the data from our animals in the yoked group, animals that didn't press any levers, that didn't make any decisions, and simply got the same amount of food and the same pattern of food for free, we saw a near identical profile. Again, dopamine levels were higher when they were getting more food, and then lower when they were getting less food. So what this suggests to us is that fluctuations in prefrontal dopamine levels seem to be keeping track of how much reward animals are receiving over time, serving as sort of a reward running rate meter. And if you think about what the prefrontal cortex might be doing in these types of situations, keeping track of actions and outcomes over time to help modify behavior, the system would need some sort of information to kind of update over a longer term period of how are we doing in terms of our profits. Are we getting more or less reward over time to help modify behavior? These data suggest that changes in dopamine transmission in the frontal lobes may be one such input to this region that the prefrontal cortex uses to help adjust behavior. In the accumbens, the story was a little bit more complicated. We again saw that dopamine levels were higher when reward probabilities were higher, and then lower when the odds of getting those rewards were less. But the profile looked quite different to what we saw in the prefrontal cortex. The first thing we noticed here was changes in accumbens dopamine levels mapped on almost perfectly to changes in the animal's behavior. So the rate of change in dopamine release over the course of the session was pretty much the same as the rate of change of how often the animal chose those risky options. Now, the other thing I'd like to point out here is you see in this profile, looking at the green circles, we see this sawtooth profile where dopamine levels are lower, and then they go up a bit, and then they're lower, or going up a bit again. What these oscillations in dopamine represent are changes in dopamine release during the forced choice portions of each block compared to the free choice portions where the animals actually have to make a decision. And as it turns out, when the animals actually have to choose, dopamine levels are just a little bit higher than when the animals simply have to make a response. Don't have any choice in the matter and just have to press a single lever. So the system seems to turn on a little bit more when the animals' actions and their choices have a direct outcome on what they might be receiving. Now, in comparison, changes in dopamine release in the accumbens did not map on very well to changes in the amount of reward they received over time, as I'm showing you in these red triangles here. But we still conducted our yoked food control experiment. Here we saw an interesting profile. What these squares are showing you here is changes in dopamine release in animals that got the exact same amount of reward and the same pattern of rewards as animals that played the task. And what I want you to notice here is that when at these points of the sessions, dopamine levels were comparable, but then the curves separated at this point here. What is special about that point there is that these are the portions of the task where the animals have to make a choice and there is some uncertainty about their action. They may not get rewarded for those choices. In the early part of the session here, they're either always guaranteed to get some reward, either a larger or smaller reward, or have no choice in the matter. But as soon as we add the situations where now the animals have to choose and they might not get a reward when they chose a certain action, dopamine levels were higher when we compare that to animals that just got this food for free. So the system seems to be more engaged when the animal has to make a choice and the animal is not entirely sure whether or not their choices might lead them to rewards. So what this suggests to us is that changes in these slower tonic levels of extrasynaptic dopamine in the ventral striatum may be integrating a variety of different signals and provide a long-term accounting of multiple types of information, including the animal's choice behavior, what it's doing, the relative amount of uncertainty associated with those choices, as well as changes in the amount of reward they're receiving over time. So fluctuations in these slower tonic dopamine levels in the accumbens seem to be integrating multiple types of information. So when we look at dopamine transmission within the striatum, there are, again, these slower changing levels of dopamine that fluctuate over longer periods, many minutes. But there are also these faster phasic signals I described to you at the beginning that are mediated by burst firing of dopamine neurons. Now, if you want to look at dopamine release that is mediated by these bursting events of dopamine neurons, other investigators have used more rapid measuring techniques that have a higher temporal resolution, such as in vivo voltammetry. And there has been a fair amount of excellent work done by a number of investigators using these faster techniques to measure rapid changes in dopamine release within the accumbens when animals were engaged in cost-benefit decision-making. So the data I'm showing you here are data from one of my good friends, Regina Corelli, and her group. And here they recorded rapid changes in dopamine release in the nucleus accumbens when animals were performing a very similar game to what I described to you here, where they were choosing between larger uncertain and smaller certain rewards. And two things they noticed here is that before the animal would make a choice, a few seconds before the animal would have to choose between the options or make a response, we would see a phasic increase in dopamine release that you can see on the time scale here lasts a few seconds rather than changes over minutes that we were monitoring with microdialysis. And the magnitude of this phasic increase prior to the choice was related to whether or not the animal was permitted to choose its more preferred option. So dopamine levels were higher when the animals were either forced to choose their more preferred option or had the choice between their preferred and less preferred option when they compared the magnitude of release when animals were forced to choose the option they didn't like as much. And then after the animal made a choice, dopamine levels increased again in a rapid and phasic manner. And in this instance, the magnitude of the change in dopamine release seemed to encode the outcomes of those choices. So if animals played risky and they won, get that larger reward, they saw a large increase in dopamine release. If they chose the smaller certain option, they saw a comparably smaller increase in dopamine release. And on those times where they played risky and they lost, they actually saw a decrease in dopamine release. Very similar to what I described to you in the beginning, where how dopamine neurons seem to encode reward prediction errors, showing a larger increase when they get an unexpected reward and showing a decrease when they don't receive any reward. So we were interested in seeing, well, how might these rapid phasic signals in dopamine release regulate choice behavior and action selection? Now, we've done our pharmacological experiments where we would block dopamine receptors, but those don't really tell you what these rapid signals are. When you go in with an antagonist and block dopamine receptors, you're blunting all types of signals, both these rapid phasic signals and these slower changing tonic signals for an hour or two. So it doesn't really parse out what specific information these phasic signals are. So we needed a way to use a temporally precise mechanism to suppress these phasic signals. And it turns out that nature has provided us with a neural circuit that exerts inhibitory control over the dopamine system, and that circuit involves a nucleus referred to as a lateral habenula. This is an evolutionary, very old nucleus. We see this in all sorts of vertebrate species. And one reason there's been a lot of interest in this nucleus is that it seems to have a pronounced inhibitory effect over the firing of dopamine neurons. Because if you stimulate the lateral habenula, that causes a marked suppression in dopamine neuron activity, very similar to phasic dips in activity that are observed when an animal doesn't receive an expected reward. Now, recordings from lateral habenular neurons in awake-behaving animals have shown that, well, these neurons, they're kind of like the opposite of dopamine neurons. I like to call them dopamine neurons from the evil parallel universe. And what I mean by that is they actually fire more when something bad happens, like the animal doesn't get an expected rewards, and they actually suppress their activity when something good happens, like they get a reward. And a number of studies over the last 10 years or so have identified that the ability of the habenula to suppress dopamine neuron activity is mediated by a disynaptic circuit where we have excitatory projections from the habenula that then target a relatively newly discovered nucleus in the back of the brain called the rostromedial tegmental nucleus, or the RMTG. And in turn, the RMTG sends a dense inhibitory GABAergic input to dopamine neurons in the VTA. So by activating the habenula, we can suppress dopamine neuron activity in the VTA in a very temporally specific manner. So we used discrete electrical stimulation of either the habenula or the VTA itself to modify and overwrite these different phasic signals that are associated with decision making, and to ask, do these outcome-related and pre-choice phasic signals modify action selection during decision making? So in one experiment, we would have a situation that where we had animals well trained in the task, and sometimes they would choose that risky option, and they would not get a reward. That's how life works sometimes. Well, what we know from previous studies that is in these situations, when they choose risky and lose, that causes a suppression in dopamine neuron activity, a phasic dip. So under these conditions, only when animals chose risky and lost, we would give a brief half-a-second stimulation to the dopamine neurons in the ventral tegmental area. The idea here is now we are overriding these phasic dips, actually causing a phasic burst, tricking the system, potentially, into thinking it may have got rewarded when it actually didn't. And what we saw in this situation is that only when we stimulated the VTA after a loss, animals started choosing more risky. They started to behave as if that risky option was paying off much more than it actually was. So nudging their behavior in another direction just by overriding these signals that occurred when the animal played risky and lost. Now, on the other hand, sometimes we'd have a situation where the animal would choose that risky option and win, get that larger reward. What we know is happening here in terms of the dopamine transmission is that that causes a brief phasic burst in dopamine neuron activity. These neurons start to fire for a brief period of time. So what we did in this situation is every time the animal played risky and won, got that larger reward, while the animal was receiving that reward, we would give a few brief pulses to the lateral habenula, which, as we know, will suppress dopamine neuron activity and suppress these phasic bursts. So here we are trying to overwrite these signals and again trick the system into thinking that maybe it didn't get the reward that it thought it did. And under these conditions, we saw the opposite of what we saw when we stimulated the VTA. animals chose the risky option less, started behaving as if that risky option was not paying off as much. They were not receiving this large reward. Even though they were tasting the food, consuming the food, their behavior, when we suppress these phasic bursts that occurred with receipt with reward, seemed to suggest that they were not registering those wins. And they adjusted their behavior thinking, hey, this is not paying off as much. Now, to be perfectly honest, we were having some fun with these experiments. Well, I was, because I wasn't actually conducting the experiments, but it was like we could remote control the animals to a certain degree. By stimulating at a certain time, we could bias their behavior one way or the other. So we did another experiment. Well, what happens when we stimulate the habenula and suppress these phasic increases in dopamine when the animal chooses that small certain option? When it goes for that safe government savings bond. So now we're simulating a crash in the bond market. So every time we suppressed dopamine activity, when the animal chose that small reward and got that small reward, we again saw a change in behavior. Here, it increased risky choice. Animals behaved as if, well, this small option is not paying off anymore. Now, it may be that these effects were due to just some aversive response. They didn't like being stimulated in the habenula, and so they were just choosing the other lever to avoid stimulation. Well, we don't think that's the case, because when we do a key control experiment, where animals are just choosing a very simple type of decision, a large reward versus small reward, both delivered at 100% certainty. And if we stimulated the habenula every time they got the large reward in this simple situation, there was no change in behavior. So it's not like they didn't like getting stimulated, but when we stimulated in situations involving uncertainty, that's when we got changes in behavior. So what these outcome-related phasic bursts and dips seem to be doing is providing short-term information about recently rewarded and non-rewarded actions to influence subsequent choice behavior. Now, what about those pre-choice signals, these increases in dopamine release that occur right before the animal has to make a choice? Well, we know right before they need to make a choice, we get this large increase in dopamine release. So what we did in another group of animals is every time they were about to make a choice, we again stimulated the habenula and suppressed that pre-choice increase. What happened? Well, one thing that happened was that it reduced the incentive salience of the levers. What I mean by that is they were now a little slower to make a choice. They didn't seem as invigorated to go after those levers, and they were more likely to miss out on trials. And in terms of their choice behavior, when we looked at the entire group, this tended to reduce choice of that large risky option. But when we probed some individual differences in our group, we found that some animals tended to just always like that risky option, irrespective of the odds. They kept choosing that risky option, whether the odds were good or bad. And in these situations, when we suppress these pre-choice phasic signals, it uniformly reduced their preference for that large reward. Other rats, on the other hand, played more rationally, chose the risky option more when the odds were good, chose it less when the odds were bad. Here we saw that suppression of pre-choice signals tended to disrupt their preference for their more preferred options. So they chose the risky option less when they preferred that option, and they chose it slightly more when they preferred the small safe options. So we interpret these data to mean that these increases in dopamine release prior to a choice seem to promote actions directed towards more preferred rewards. Now, dopamine doesn't seem to be involved in actually making the preference, in deciding what's the better option. This is mediated by these other circuits I've been talking about, the frontal lobes, the amygdala, what have you. And dopamine helps ensure that once a certain bias has been established, that the animal's behavior is directed towards what is deemed the better option. Now, lastly, I want to finish up with some studies looking at the involvement of the lateral habenula itself. Because while stimulation has been a useful tool for manipulating phasic signals, we wanted to see, well, what happens when we remove this node out of the circuit? So for those of you who are unfamiliar with the anatomy, I'm showing you here that the habenula is a relatively tiny nucleus, and it's somewhat difficult to hit surgically. So my student who worked on this, Colin Stopper, he initially did a set of experiments, and he came to my office after he completed, and he said, Stan, there is no effect on choice. And I said, well, let's check the histology. Turns out there were a bunch of lovely placements in the dorsal hippocampus, right above the habenula. So we don't need that part of the brain to guide decision making. I said, try again. Came back a few months later. There's still no effects now, and I looked at the histology. Now we're getting closer to our target, but still no effect. Try again. Another experiment, now he overshot. Now he was below the habenula, still no effect on behavior. And we were getting a little frustrated at this point because it's like, what does it take to hit this nucleus? Now finally, after about a year of trying, my student was able to amass enough placements within the target nucleus. And what did we see? Well, rather simple-mindedly, we thought that if the habenula is telling you what's bad in the environment, then if we inactivate this nucleus, we might have a situation where animals just don't recognize that they're losing, and they keep choosing more risky. But we saw something much more profound. When we inactivated this nucleus, we completely abolished any decision bias they had. They went from choosing rationally, choosing risky more when the odds are good and choosing less when the odds were bad, to random and haphazard patterns of choice. So that as a group, they were choosing both options roughly 50% of the time, behaving as if they had no idea which was the better option, and so they were choosing completely random. Now, whenever you see an effect of this magnitude, well, maybe they're just blind. Maybe they don't know the difference between their left paw and their right paw. Doesn't seem to be the case. Because if we give them a simple decision, big reward, small reward, both delivered with 100% certainty, and we inactivate the habenula, there was no choice. When the animals have to choose between two rewards that differ only in their objective value, big versus small, the habenula doesn't seem to be involved. But as soon as there is some sort of cost, where the animal has a subjective preference it has to use to guide its behavior, the habenula seems to play a fundamental role in expressing subjective choice biases. So when we integrate these last bits of information in our model, seeing that these slower changing tonic dopamine levels in the prefrontal cortex and the accumbens seem to have a long-term accounting of choice and reward history. Prefrontal cortex dopamine tracks changes in reward rates over time, whereas accumbens dopamine, the tonic signal, integrates information about the amount of reward received, the relative amount of risk, and choice. In contrast, these phasic rapid signals seem to play a shorter-term accounting, conveying short-term information about the outcomes of choices and then promoting actions directed towards more preferred rewards. And within this entire circuit, the lateral habenula seems to be a key linchpin, allowing these cortical, striatal, limbic systems and the dopamine systems to work in a coordinated manner. Because when we suppress activity in this nucleus, it throws the entire system in disarray. Animals don't know what the better option is and behave randomly, suggesting that this evolutionary old nucleus plays a key role in enabling expression of subjective choice preferences. And with that, I would like to thank the great students I've had over the years. I've had a number of talented, hardworking, as you can see here, photogenic students. Those who are shown in the pictures here are those who contribute directly to the experiments, along with a number of other students I've had over the years. And this is a recent shot of my wonderful lab here, and I'd like to thank you all for joining me online. I will now take any questions.
Well, thank you, Dr. Floresco, for your fantastic presentation. That was really, really informative, and I learned so much. So as a quick reminder for our audience on how to submit questions, simply type them in the Q&A box, which can be found in the green Q&A button at the lower left of your presentation window. We'll answer as many questions as we have time for today. So the first question, while we're waiting for our audience members to formulate their questions, if I could ask a question, could you comment on how dysfunction in the dopamine circuits relate to psychiatric disorders that we see in that affect human behaviors?
Of psychiatric disorders that seem to be associated with dysfunctions in the dopamine system. And so one thing that our data showed, that, and we are building on a vast literature of people who've looked at this type of work, is that dopamine activity seems to signal important events in the environment and may increase the likelihood of an organism engaging in similar behaviors that lead to this increase in dopamine. And so if we have a situation where we have excessive increases in dopamine release, like we might see in drug addiction or in schizophrenia. This aberrant increase in dopamine transmission may, in the example of addiction, may promote individuals to continue to engage in drug-seeking behavior in response to cues or what have you, and allow them to, or kind of make it harder for them to stop engaging in these types of drugs. With respect to schizophrenia, we know that dopamine seems to be related to the delusions and the psychotic symptoms, and excessive increases in dopamine activity may be involved in kind of having an aberrant salience attribution, where individuals pay more attention to seemingly innocuous stimuli, and that may contribute to their delusions. On the flip side, reductions in dopamine transmissions may be related to certain aspects of depression, where individuals might not have the energy or the motivation to go after good things in life, which is a key symptom associated with depression.
Great, thank you for that. So we're running a little bit short on time, so I would like to once again thank you for such a fantastic presentation. Do you have any final comments for us on how these neurocircuits affect human behavior and decision making?
Forward with these types of experiments. What I think, we need to understand, we love to kind of black box different brain structures. This brain region does that, this brain region does that. But I really think as we continue with behavioral neuroscience and understanding how the brain controls behavior, we have to think of it in terms of dynamic interactions between different systems. And one brain region may contribute in a variety of different manners to control behavior, depending on the different inputs and outputs it's connecting with. And if we want to understand what's going on when the brain doesn't work well in psychiatric disorders, I think we need a firm foundation in seeing how the brain normally works to solve problems. That gives us greater insight into neural dysfunction.
Okay, that was fantastic. I'd like to once again thank the audience for joining us today and for their interesting participation. We have some questions that we didn't have time for today, but we will try to address those by Dr. Floresco via the information that you provided at the time of registration. This webcast can be viewed on demand through September of 2016. LabRoots will alert you via email when it's available for replay, and we encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.